Hello everyone, welcome back to my YouTube channel. In this session, we will be discussing about the transfusion reactions are an example of hypersensitivity too. So first we know the transfusion reaction. So what is the transfusion reaction? Transfusion reaction are hypersensitivity reaction that can occur when incompatible blood is transferred into recipient body. So this reaction can range from mild symptoms such as fever or itching or to severe complication including hemolysis. So hemolysis it is the destruction of red blood cells. Now the second, that is anaphylaxis. Okay, this is a severe complication example. Now anaphylaxis that is a severe allergic reaction or even death. So now here we understand transfer reaction in the deep. So several proteins and glycoproteins on the membrane of the red blood cells are encoded by genes with the several allelic forms. Okay, so here it is the word allelic forms. So what is the allelic forms? Allelic forms refers to different version of gene. Okay, you can say the alleles that exist at the same location, that is the locus on a chromosomes. So in the context of blood group, allelic form represent different variation of specific genes that encoding blood group antigen. Right? Okay, this is this is a glycoprotein. Okay, so this is RBC cells. Their surface containing, that is the protein, the carbohydrate chain. Okay, so the protein and carbohydrate chain. Now come to the allelic forms. Okay, so for example, in the ABO blood group system, these are three main allelic ABO. Right? So this is the alleles of A, B and O. So these alleles determine the presence or absence of A and B antigen on the surface of red blood cells. Now, so even individual inherit one allele from each parent, right? Either A or B. Result in different combination of alleles, thus different blood types. Now, interestingly, the blood type antigen ABH are carbohydrate rather than the proteins. Okay. So this is a protein, but here it is the carbohydrate chain. So this is a work as the antigen. Right? Now, so how they define, okay, blood antigen? It is the carbohydrate rather than the protein. So scientists, okay, they demonstrated by simple experiment in which the addition of high concentration of particular simple sugar was shown to inhibit antibody binding to red blood cells. Okay. Now the okay, here. Suppose here you can see this is a antigen, B antigen, and H antigen. Okay. So H antigen. Okay. H antigen is present in all blood types. Okay. So this in this experiment, they was discovered that the blood type antigen are carbohydrates rather than protein. Okay. So the simple sugar, like they added the simple sugar like fucose, suppose. Not fucose, because this is H antigen. Okay, this is the H antigen structure which is present in all blood type. Okay, so here it is the H antigen structure. Okay, here it is the also H antigen structure in all blood group. Right? Okay, so they added the high concentration of particular simple sugar was shown to inhibit the antibody binding to red blood cells and bearing particular type of red blood cells antigen. Okay, that sugar, they added this sugar, they work as the blood type antigen. They binds with the antibody, okay, rather than the red blood cells surface antigen. Right? Now, so this inhibition reaction revealed that antibodies engaged to group A antigen. Here it is, okay, mostly bound to N-acetyl glucose amine residues. Same antibody engaged to group B, okay, group B antigen bound to galactose residue. Here it is the galactose residues. And third, that is the antibody engaged toward the H antigen bound to the fucose residues. Here it is the fucose residues. Okay. So the H antigen present in all the blood types. Okay. So this figure indicate the structure of terminal sugar of blood group, which constitute unique epitope of the A, B and H. A, B and H antigen. So it is the word that is the epitope. Okay. Epitope are the part of molecules such as protein or polysaccharide that can be recognized by the antibody. Okay. And or immune cells. Okay. Then that triggers the immune response. Okay, so the A, B and H antigen are synthesized by the glycosyl transferase enzyme. Okay, so what is the glycosyl transferase enzyme? It's which is type of enzyme that catalyzed by the transfer of sugar molecules or sugar residue from donor molecules to specific acceptor molecule. For example, H antigen. Okay, so the glycosyl transferase, which plays the transfer of sugar molecules from H antigen to A antigen of the end site glucose amine. So this enzyme play a key role in the biosynthesis of complex carbohydrate, glycoproteins and glycolipids. Okay. Now the ABH. Okay. So now the antibody fixed toward the ABH antigen are termed as the isoheme agglutinase. Here it is. Okay. So this is the table show the pattern of blood antigen. Okay, and express the isoheme agglutinase normally found within the human population. So first that is a genotype. Okay, ABO genotype, corresponding blood group phenotype. Now the antigen or erythrocytes agglutinase. Okay, and serum antibody. Okay, isoheme agglutinase. Okay. Okay, so this is the how this for example blood group phenotype A, so antigen A and antibody B. Right. So this is the picture. Okay, red blood cell type. Suppose this is a blood type AB O. Right. Now the isoheme glutinase antigen on red blood cells. Right. Anti-B if the person has the anti-B if the person has the blood group A. Okay, so this is the A antigen and anti-B. Now we understand the B cell generating antibodies undergo negative selection. Okay. So many adults have IgM antibodies targeting specific members of the ABH blood group system that they themselves do not possess. Okay, here we take the example. So imagine Sarah has blood type A. Okay, Sarah has blood type A. In her body, she naturally produces the antibody against the B antigen. That is they produce the antigen against the B blood group. Okay, so B antigen. Now, it's part of our immune system's defense mechanism. Okay, this is the part of. Okay, however, Sarah does not have the B antigen in her own blood. Okay, because she has blood type A. Okay, so this is a type antigen also present in her body, but B not. Right. Okay. So her immune system produces the IgM antibody. Okay, so this is a particular IgM antibody. Okay, IgM is targeting the B antigen. Okay, even though she doesn't express itself, okay, they naturally produce the antibody IgM against the B antigen in her body. This is because common microorganism. Okay, so this is the also this common microorganism. Okay, express carbohydrates antigen very similarly in structure to the carbohydrates of the ABS system and induce the B cell response. Okay, so here we also take the example. For example, individual with the blood type A, okay, this Sarah, okay, recognize B like epitopes on microorganism. Okay, suppose same like, okay, same like B antigen, blood type B antigen are also expressed on the microorganism cell membrane, okay, and produce the antibody to the B like epitope. This is not totally B, okay, this is similar, 100% similarly blood type B antigen which are emerge into the microorganism surface. Okay, so the Sahara body or individual body produce the B like antigen antibody. Okay, so however, in this same individual, okay, does not respond to A like epitopes on the same microorganism because they have been tolerized to self A epitope. Okay, so in simpler term, this means that the person mentioned previously does not produce a response to A like structure on a certain microorganism. If the person has the A epitope, okay? Because the individual produce the B antibody against the B epitope. Here it is the twist. For example, okay, so this okay, individual they produce the not produce the A antibody because they have been tolerized to self A epitope. Okay, so here we take the example. Let's go back to the Sarah with blood type A, right? So her immune system does not react strongly to A-like structure on certain bacteria or viruses because it recognizes them as the similar to her own blood type. Now, it's like her immune system has been trained to ignore this structure, treating them as a part of herself. Okay, so if the individual body has entered the if they enter the microorganism which have the A epitope on the surface, but the body they ignore this structure and treating them as a part of herself or individual cells. Okay. Okay. Now the B cell genetic antibody specific for the ABH antigen expressed by the host, however, undergo negative selection. Okay. So this is how the negative selection. Suppose if type A individual is transfused with the blood containing type B cells. Okay. Now you are getting. Okay. So A individual is transfused with the blood containing type B cells. Okay. So transfer reaction occurs in which the pre-existing anti-B, okay, anti-B antibody binds to the B blood cells and mediate their destruction by means of complement mediated lysis. Okay, so now okay, let's break it down with the simple example. Okay, so for example, individual with blood type A recognize the B like epitope microorganism and produce antibody to the B like epitopes. Okay, so this is same individual does not respond to A like epitopes on the same microorganism because they have been tolerized to self A epitope. Okay, so this is I have been discussed. Okay. Now here it is we take the example. Okay, imagine there is a person named Sarah who has blood type A. Sarah has immune system is like protector that fight off invaders like germs by making special protein called the antibodies. Okay. Now let's say Sarah's immune system recognizes some germs that look a bit like blood type B. So her body make antibodies called the isoheme agglutinase to fight those germs. Right? They produce the antibody B. Okay. Now, if Sarah ever gets a blood transfusion with the blood containing type B cells, her immune system might think those cells are invaders and attack them. This is because Sarah's immune system already has those antibody ready to fight off anything that looks like blood type B. Now, here is the twist. Sarah's immune system knows not to attack her own blood type A. It's like her immune system has learned to ignore her own blood type characteristics. Okay, so second, individual with the blood type O express only the H antigen, as we know. Okay, so this person, they can donate blood to anyone. They have antibodies that will react to both A type and B type blood. Their anti A and anti-B producing B cells were never exposed to A or B antigen and therefore were never deleted. Okay. So anti-A and anti-B producing B cells were never exposed to A and B antigen. Let's break it down with the simple example. Okay. Imagine a person named John who has blood type O. People with blood type O only have the H antigen in their blood. Okay. Now, John can donate blood to anyone because his blood doesn't have A or B antigen. Okay, which means it will not trigger reaction in the recipient's body. Okay. However, John's immune system has antibodies that will react to both A type and B type blood if they receive them. Okay. Why? This is because his body has never been exposed to A and B antigen before, so his immune system has not learned to ignore them. So here is the interesting part. Zone immune system has B cells that produce the antibodies against both A and B antigen, and these cells were never exposed to A B antigen, so they were never deleted from his immune system. Okay. So in simple terms, even though John can donate blood to anyone because of his blood type O, okay, if he receives blood with the A and B antigen, his immune system may react to them because it has not learned to recognize them as a safe. Okay, so this is all about today's video. I hope this will helpful. Okay, thank you for watching.